Nu ska vi se. Där syns jag, ja. Okej, så välkomna allihopa till dagens disputation. Som jag tänker vi helt enkelt kör på engelska från första början. Funkar bäst för alla. So welcome to today's dissertation where Björn Forsberg will defend his thesis entitled Fast and Reliable Alignment and Classification of Biological Macromolecules in Electron Microscopy Images. Very good. So my name is Pia Avelot, by the way, and I'll be sharing this event. These are unusual times. So today we have the opponent and all of the committee with us online. And the Faculty of Sciences here at Stockholm University have appointed Professor Sriram Subramaniam from the University of British Columbia to be the faculty opponent for Björn's thesis. And we're very grateful to you that you could join us today. I think you are there. It's a very important job, and I know you had to get up really early in Vancouver, right? I think, to do this. In the committee, we have Professor Suparna Sanyal from the Department of Cell and Molecular Biology at Uppsala University. We have Professor Xiaodong Zhu from the Department of Materials and Environmental Chemistry here at Stockholm University. And we have Docent Osan Öktem from the Department of Mathematics at the Royal Technical University. And thank you all for joining us. And I hope you can hear us and see us. And you're all here. Very good. So the procedures today will be that first, Björn gets to announce if he has found any errata in his thesis. Then the opponent will give a brief introduction to the field of the thesis and try to put it into context. Björn will get to summarize his main findings briefly, and then we will take a short 5-minute break just to stretch our legs. And before we start the actual questioning and discussion, first with the opponent and then with the committee. And then just as usual, this thesis is open to questions from the public, either the three of you in this room or the more substantial amount of people online. So, and those of you who are online, I will ask you to put yourselves on mute. Otherwise, Erik and I would put you on mute. But if you do want to ask or say something, either raise your hand or indicate it in the chat or something so that we know. After we have had all the questions that have to be asked or want to be asked, we will close the open part of the dissertation. And then we will meet with the committee and with Björn's supervisor, Erik Lindahl. And technically we'll do that in a different room. So those will leave this room physically and online, and we will meet in another room. And we will decide on we will decide, I will have no part in that decision, but the committee will decide on the grade, which is either pass or fail. I have no other grades here in Swedish education system for dissertations. And we will then come back physically to this room and online to this Zoom room to announce the decision. With that, we are ready to get started. Those of you who are in here, please put your mobile phones on silent. And also what is really important for this dissertation is that the committee and preferably the opponent can follow everything that happens in here. So they have to indicate somehow. They can call me, they can indicate in the chat or something if there's any problems. And then I have to interrupt the proceedings until we get it fixed. Just so you know if there's any interruptions. Yes, I think we are ready to get started. Björn, any erratas?
Yes. So let me just bring that up. So hopefully you can now see the corrections that I want to amend with my thesis. So first of all, there should be a list of papers at the very beginning of the thesis itself. That is missing. So But from page 81, you will find a summary of papers, which also lists the papers. In addition, there are some minor mistakes that I've noted. On page number 1, it is stated that the first protein to be determined by X-ray crystallography is hemoglobin in 1958. This is, of course, myoglobin, not hemoglobin. On pages 15 and 17, I use the term spatial resolution, where spatial frequency might be more suited and what was intended. On page 60, there's a statement referring to k equals 0, which should be n equals 0. And reference 61 was accidentally made to an erroneous paper. The reference should be to the paper High Resolution Noise Substitution to measure overfitting and validate resolution in 3D structure determination by single particle electron cryomicroscopy. The paper referenced is a paper on a similar topic from the same first author, hence the mistake.
Oh, I'm so sorry. Yes, thank you very much, Björn. So, Björn, do you want to share your presentation with us?
Okay, will do. Can you see it now?
Yes.
Okay.
Yes.
Okay, well, shall I get started, Pia? Is it good now?
Yes, please, please. Thank you very much.
Yeah, good afternoon, all of you. In Stockholm, it's very early in the morning here in Vancouver, and I'm really honored to be the thesis opponent for Björn Forsberg. I wish you all the best, Björn, today in your exam. I just want to especially thank you for having this in the afternoon and not in the morning in Stockholm time. I'd like to provide an introduction to the field of cryo-EM, which is the subject of Björn's thesis. I'd like to begin with a general overview. I understand that the audience is broad, so I've kept this fairly general in terms of the principles of the field and how it might connect broadly to technical and biological aspects that are relevant to today's discussion. Okay, my screen seems to be frozen. Let me see if I can. Okay, it's a bit of a technical issue here. Bear with me just a second. Okay. I'm not able to advance my screen while I present. Let me make sure I can get this straight. Can you see the first slide now?
Yes.
Yeah, okay. If you look at the spectrum of sizes in biology ranging from small to large, over the last decades, many decades, there have been developments in imaging that span the gap all the way from looking at molecules to very large objects such as humans in biology. And this spans a size range of about 28 orders in magnitude. And historically, X-ray crystallography and NMR spectroscopy have been useful both at the left end and the right end of the spectrum to look at small molecules by averaging and getting distance constraints, and large entities such as mice and humans by imaging methods. And in the middle, the realm of microscopy, both electron and light microscopy have historically played a great role in unraveling biological complexity. The area that we care a lot about at the junction between structural and cell biology is roughly this on the size scale of viruses. And for many years, for me, one of the orienting sort of ideas in why we care about imaging is to understand phenomena such as the one shown in this movie that was taken over 60 years ago, where we're looking at the interaction between a human neutrophil on a petri dish and a bacterium. And clearly, this battle of the immune system fighting a foreign invader is played out in our bodies all the time. And while a movie like this captures the basic idea of a cell interacting with another cell, we like to understand this at a molecular level. So there's quite a number of molecular machines that obviously have to be at play for a process like this to be successful, either for the invader or for the cell that's defending the body. And this really is the subject of structural biology that is very closely connected to how cells work. And this really is very close to the discussions that we will be having today. Several years ago, David Goodsell, who is at Scripps, pointed out that it's actually useful to be able to visualize the general architecture of cells at the right scale, where we might place things into the context. And he drew a number of diagrams like this, which were very helpful in providing a perspective to the general audience in terms of what cells might actually look like if you place the ribosomes and the nucleoid and various membrane proteins in this instance in context. And I want to share with you a short paragraph that he wrote in 1991, a long time ago, where he said that a clear picture of the interior of a living cell that shows the average distribution of molecules at the proper scale, proper concentration, and with no missing parts seems to be central to the understanding of the workings of life. However, this type of picture is virtually absent from the popular and technical literature. And he goes on to say the reason is simple, and I'm now reading the part that's underlined in red, which is that electron microscopy gives a view that is too coarse. Subcellular structure is studied, but individual molecules are not seen. Fortunately for us, that turns out to be incorrect. And as all of you know, just a couple of years ago, the Nobel Prize in Chemistry was awarded to Richard Henderson, Joachim Frank, and Jacques Dubochet for really showing that one can transition out of this stage of looking at things to be too coarse to actually getting to molecular structure. So today I'd like to, in my short presentation, touch on three topics. I'd like to provide a brief introduction to the evolution of cryo-EM technology or cryo-electron microscopy technology, a couple of examples to imaging molecular machines similar to the ones that our candidate today will speak about, and finally, something that's very topical, the relationship of cryo-EM to viruses, and in particular, COVID-19, just to bring us up to date with why these technologies are particularly relevant in today's world. I'd like to begin with a description of the technology itself. The way that objects can be imaged with an electron microscope is fundamentally similar to what can be done with the light microscope, in that there's a source of radiation, which are electrons, a specimen, a lens that is more complex than what's shown here, but nevertheless achieves the same effect of focusing the radiation both in an image plane and also collecting it in a diffraction plane. This idea of imaging with a lens is not new, of course. We have Van Leeuwenhoek, who came up with the original original advance in this field, and you can see him at the left looking at something that he picked up in his pond. That is at the back of this handheld microscope, which is about a couple of inches long, where his source of radiation is a candle. You can see the lens is one that he ground himself, and the imaging plane is his eye. You can see his pupil, in fact, is slightly dilated. So this is the original microscope. And one of the interesting aspects here is that you can see that he is very much larger than the microscope. On the right-hand side, you see a modern electron microscope that we use to do cryo-EM. This actually was the very first commercial Titan Krios microscope. We took delivery 10 years ago at the NIH, where I was until recently. This is the real size of these people. These are not particularly short people. It gives you a feeling for the size, in that the relative ratio between the microscope and the experimenter now has reversed, and the microscope is quite a bit larger than the individual carrying out the experiment. These advances in cryo-EM that lead to work such as what we'll hear about today can be summarized in these few slides. The most exciting aspect of these changes has been that it is now possible to describe structures of macromolecules, proteins, nucleic acids at high resolution, but without the need to crystallize the protein first. And this is a technology that crystallography has been around a long time, but the presumption that you need to crystallize it to get to atomic resolution is now no longer true. In particular, the application to dynamic protein assemblies is very relevant. Cells work, as you saw in the animation before, by movement, and the use of these methods to describe and capture snapshots of motion is very useful in a mechanistic understanding of how cells work. It's also become very, very useful to look at the conformational states, particularly of integral membrane proteins, which have historically been difficult to study using classical structural methods such as crystallography and NMR spectroscopy. And here again, the value of cryo-EM has been extraordinary in providing insights that have not been possible before. In the context of its application to society, there's major interest now in using these technologies to map drug binding sites to understand better how drug molecules work. And in that, in order for that transition to take place, it was necessary for the technology to achieve high resolution. And once again, that has now happened. And that's also another very exciting area going forward. And finally, all of this has been made possible by a very high degree of automation in data collection and processing. And we will be hearing about that today from Björn as he presents his work. A few years ago, cryo-EM was listed as a method of the year in the cover of Nature Methods. At that time, we had just reported the structure of beta-galactosidase, and this essentially was about the only time that we see an intersection here between global warming and cryo-EM that sort of captured in this cover a few years ago. But the important point here is that this actually wasn't really new at the time. Cryo-EM has been around for quite a while. And if you look at the history of this field, this is now from the EM data bank yesterday. The number of entries in the EM data bank as of today exceeds over 10,000 for maps deposited. But if you go back to 2016, which is right about here, you can see the field was already growing quite rapidly at that time. There's a lot of work at various resolutions. But the thing that was missing until 2016 was the number of entries at atomic resolution. And this, there were a few examples from electron diffraction, but I think the transition point really was the ability to obtain these kinds of structures at atomic resolution, which is really where we care about the chemistry of how proteins work. Cryo-EM, on the one hand, looks very easy. Many of you perhaps have had direct experience with it. Here's an animation from a while ago that we made in terms of how it works. It begins with a drop of solution that contains a protein of interest, or perhaps a virus or a cell of interest. This is deposited on a microscope grid that's about 3 millimeters wide and is blotted by Whatman #1 filter paper that many people are familiar with from chemistry. And at this point, what's left behind is a thin film of liquid. And plunge freezing this specimen in liquid ethane that's cooled by liquid nitrogen vitrifies the sample and essentially freezes the motion of the proteins, but hopefully captures it in multiple orientations. The delivery of this frozen specimen at liquid nitrogen temperatures into the microscope then results in the next stage of the process, where data can now, as a result of major innovations in hardware, be collected automatically. And the unit of information is either a micrograph, which captures it's a single shot, or a movie that captures essentially the images as the protein is being exposed to, as the specimen is exposed to electrons. Once the data is collected, it initiates a series of processes where one can then cut these molecules up computationally, and if all goes well, convert this into a 3D map. And this step that shown here in just a few seconds is the subject of a good portion of today's thesis presentation. So some years ago, when we were working on beta-galactosidase, we essentially were able to show that from the images like this, it was possible to get to resolutions where we could visualize the side chains. And this was very exciting because it showed that we could now begin to describe structures of proteins such as beta-galactosidase without actually having to crystallize them. And in particular, the excitement was that we could visualize not just the side chains, but also visualize water molecules, which are interesting, not just mechanistically, but particularly relevant for drug design and other applications where very precise information is necessary to understand exactly how various compounds work in inhibiting or activating protein function. Over the years, things improved. A few years ago, the level of detail that we had in the side chains was what we thought was good at that time. But now clearly with further improvements, we can now essentially visualize the contours of individual atoms simply by imaging and averaging tens or hundreds of thousands of projection images of single molecules. And this really now places us in position to be able to take on interesting and important biological challenges. The fact that we can see these side chains at atomic resolution is exciting from the viewpoint of chemistry, because we now no longer have to guess about exactly what the orientations of these side chains are. And in many cases, for proteins that are dynamic, this is difficult to achieve simply because there's motion and there's multiple conformations. But there are several regions in protein assemblies, and maybe we'll hear about this today, where they're sufficiently well ordered that one can actually average to get information at this level of resolution for essentially all side chains, small and large. I'd like to switch now to presenting a few examples of how this technology is useful. In biology, I'd like to give 3 examples, all perhaps relevant to today's discussion. And I want to highlight that one of the major advances that took place in the field over the last decade or so is the switch from looking at what used to be called traditional cryo-EM targets, typically large viruses or really large protein assemblies such as the proteasome, the ribosomes, to looking at much smaller proteins and much smaller complexes, particularly protein complexes that move and change conformation in the course of their normal activity. And so this transition from looking at large to small and dynamic really is one of the major changes that have taken place in the field over the years. The 3 applications I want to discuss today are relevant to the dynamic aspect of proteins. The first of these is a protein complex called pyruvate dehydrogenase, which we worked on beginning about 2 decades ago. And the idea was that if you were to look into a cell and looked at all the reactions that are taking place, you would get a feeling for the metabolism of the cell and the molecules that are produced, the proteins involved. And over decades of biochemical analyses, we now have a good idea of the proteins that are involved in these functions. So if you just shut your eye and blindly pointed at something in the middle, there would be a protein that perhaps some biochemists had purified. And our interest, and that of the field at large, is to understand how these molecules come together in 3D. If you were to walk into the cell, you wouldn't see a chart like this, but you would see something in 3D. And this is work that we did, beginning with a collaboration with Richard Perham at Cambridge, which was to understand the architecture of a complex called pyruvate dehydrogenase, whose job is to produce a molecule called acetyl-CoA. All organisms have this enzyme complex. It's about 4 times the size of a ribosome. And our interest really was to figure out the mechanics of this machine. Where are the moving parts? What is it? How does it actually work? And the main discoveries that came from using classical cryo-EM methods was the discovery that these moving parts are largely confined to this annular region that you see, and that the conversion of pyruvate to acetyl-CoA takes place in a couple of discrete steps, where each flash is one reaction in this process. Essentially, this is the kind of information that we thought would be possible, where we relied on architectural information, but used information before from crystallographic constraints to derive mechanism. This is a recent example from work we did last year, where we can now transition from these kinds of architectural descriptions to molecular descriptions in great detail. Here are three steps in a reaction that's carried out by Cas9. Many of you no doubt have heard of this. This is CRISPR-Cas9 technology is very much in the news in terms of its relevance to gene editing and the prospects for improving human health in this century, I would say. What we care about here is exactly the specific event where the DNA is cut by Cas9. And using cryo-electron microscopy, we showed that this remarkable process where that we call the Cas9 scissor involves very large motion of a portion of the enzyme, where there's a huge change where it comes from far away and essentially executes the cut. And we know that it's cut because if you look on the left-hand side, you see the phosphodiester bond is intact. But if you look right after the cut, the bond is gone. So the fact that we can capture dynamic states like this is really very powerful, is a powerful use of cryo-EM technology. The last example I'd like to share with you involves drug discovery and how these technologies might be useful to map drug discovery. This is the example of an enzyme in cells that has, it's a protein called P97. It has 3 distinct domains, an N-terminal domain, and 2 nucleotide binding domains. And using cryo-EM, we were able to show that these and perhaps many more states are populated in the course of a catalytic cycle. But what we care the most about in this context is, despite these kinds of motions, to be able to capture how small molecules bind and capture these at atomic resolution because they may trap particular states. So these kinds of studies were unthinkable a few years ago, but the fact that we can now execute them at high resolution is indeed suggests great promise for this technology in the years to come. In the last few minutes, I'd like to touch on the application of cryo-EM to infectious diseases, in particular viruses. And I'd like to introduce this using one of the viruses that we had worked on many years ago, HIV, which is where there's no vaccine yet, with well over 30 million people in the world infected with HIV, leading to AIDS. If you image a virus such as HIV with cryo-EM, where we look at the structures of individual viruses, one of the remarkable things is that these are called envelope viruses, where each virus particle is differently shaped, differently sized, has varying numbers of these little blips on the surface, which are called the envelope glycoprotein spikes. These are membrane proteins that stick out, and that's the only protein that HIV needs to get into cells. On the right-hand side, you see a reconstruction of this spike from electron tomography, and this is a variant of cryo-EM. Perhaps we'll touch on this in the discussion, where instead of imaging specimens only in projection, it is possible to collect a series of images by tilting the specimen relative to the electron beam. This is a bit like a CAT scan that any of us might have had, where you sit in a spot and the beam moves around you. Here, the specimen moves, but the beam is fixed. But as a result of this, what comes out is a density map that contains information on the 3-dimensional architecture, in this case of a single HIV virus. And then once we have this, one can then take this information in 3D and computationally tease this out by essentially searching the surface of this in a bank of computers to select what look like these spikes. And years ago, a decade or so ago, we showed that you could take this information and determine the architecture and structures of these proteins to stick out of the viral membrane. And information like this has been and will be very useful for vaccine design. It's also important, this kind of information ended up being extremely useful to map antibodies that bind to HIV and viruses like this, to define exactly where they bind and whether they block the entry of these viruses into cells. And this kind of information, again, is it's critical to understand how exactly one should go about finding therapeutic treatments and vaccines for viruses such as HIV. In similar work, we extended this to looking at Ebola and to influenza. There's a lot of fantastic work in the world on looking at using these technologies to look at how viruses work and how one might actually find ways to fight infections caused by these viruses. The last example I'd like to share with you is work on COVID-19. 3 groups across the world very quickly moved in February and March to generate structures of the proteins that sit on the surface of the membranes of these. On the left-hand side is the COVID-19 virus. Right-hand side is a cell that has a receptor for it. And this is a movie downloaded from the EM data bank, where they show that indeed we now, literally in a couple of months, we have an understanding of the interaction between the receptor on the human cell and the viral protein that the virus uses to get into cells. And information like this clearly will be very relevant to understanding how to block the entry and hopefully to also design vaccines. So I'd like to conclude by pointing out that as the technology has evolved, it has become a revolution. And we now can look at structures, not just in terms of blobs that you see on the left, but in atomic detail. And we will hear more about this today from our candidate. And I'd like to finally end by noting that the word cryo-EM is indeed a broad discipline, while the presentation I've given right now, and perhaps the discussion today, will focus primarily on the use of this technology to look at proteins, its relevance to looking at viruses, cells, and tissue is also likely to be very important in the years to come. And maybe we'll pick up on this in the discussion. Anyway, that's all I have to say. Thank you very much. And I look forward to hearing Björn's presentation.
Thank you very much for that nice introduction. Let's see, Björn, if you can connect. Very good. Okay.
One second. I still
Why
have
don't
to
you summarize the last 5 years in 20 minutes?
Yes. 1 second. So I believe everybody can see my presentation now. Is there anybody who cannot? Sorry. Yeah. So today I will be sharing some of my research over the last 5 years relating to the three-dimensional reconstruction of biomolecules using cryo-EM, and specifically the implementation of some fast algorithms to do so, and the methodological implications that we saw in some practical projects working with this as well. So I will start out, I will cover the 5 papers included in my thesis, which will start out method focused and then progress towards more of an application focus. But this will always keep a mind towards the methods. So as Sriram described, I will briefly just recap that cryo-EM tries to visualize extremely small biological molecules or biological building blocks in general, made from proteins and or DNA or RNA and so forth, by suspending them in a thin sheet of ice and transmitting electrons through that sheet. What we end up with is so-called micrographs, which are transmission images of these molecules. Each biomolecule is then denoted a particle and consists a 2-dimensional projection image of the original 3-dimensional object. The aim of cryo-EM now is to reconstruct that 3-dimensional particle to infer the biological information. The computational challenges here are then to find the missing information that we need in order to do this reconstruction. Principally, we need to figure out the orientation of each particle in the ice, because this is presumably random and preferably actually random. But there are 5 such orientational parameters to be determined for each particle. And keep in mind that we have hundreds of thousands or even millions of particles to process. At the same time, we also need to be vigilant that not all particles necessarily stem from the same underlying structure. So we need a method called classification in order to group our particle images into homogeneous subsets that represent this same underlying entity. Tackling both of these computational challenges at the same time creates a fairly substantial, if not enormous, computational challenge that up until a few years ago was really limiting the method's application. So a single run of alignment and classification for a reasonably sized data set could in fact demand large-scale computational resources for multiple days, if not a week or more. Nonetheless, as Sriram pointed out, the transformation going from this 2-dimensional projection data to the 3-dimensional reconstruction is really worth the effort, because it affords a wealth of biological knowledge we can infer information about the structure and dynamics of proteins and other biomolecules. We can start to map their interactions and even understand the chemistry that goes on in biological molecules. So it is in the interest of cryo-EM to make sure that we can do as much of this reconstruction as possible. Speeding up the algorithms is, or was, a primary focus for us in this case. So what we did was we teamed up with Shores, the originator of the Reliant software in Cambridge, to look at the computational bottlenecks of Reliant. What we did there was to restructure the algorithmic workflow and expose parallelism so that it could be offloaded to GPUs. So GPUs is a specific type of hardware that is extremely well suited to do parallel computations and image processing, which is the bulk challenge of this reconstruction processing cryo-EM. What we were able to achieve with that is a 10 to 40 times faster alignment and classification method. This has really made cryo-EM much more accessible and made the hardware necessary much more compact. This means that you can also reprocess your data more and more extensively examine the data that we collect to better infer biological knowledge. Of principal importance as well is that it has allowed additional methods, more complicated methods or demanding methods to be developed and checked in full. The same paper also includes some an even greater speed up to a step called particle picking, that is the isolation or identification of particles. This again is just an example of how faster processing affords better interpretation, because you can now rerun your analyses until you see that the results are optimal. Paper 2 was really about taking the developments of paper 1 and making sure that we didn't require GPUs to see a good utilization of the available hardware. So making sure that you could run Reliant in an efficient way on, for instance, supercomputer resources that don't have access to GPUs. There's also a number of different processing challenges, namely large viruses, for instance, that tend to create large enough reconstructions that they cannot fit in terms of memory onto GPUs. So this was really about making sure that we get maximum accessibility of the Reliant methods on any hardware. The speed up, as you see, is much more modest, but you have to keep in mind at this point that this is now comparing previous versions of the same software running on exactly the same hardware. And in that capacity, this is really a substantial speed up. The paper also included a number of additional measures that increase the interpretability of the final reconstructions. This is centered mostly around better correction estimations and a number of different features for special consideration at high resolution. But this has really helped us to understand what limits the available resolution and how far we can take cryo-EM data. So to briefly summarize paper 1 and 2, we've been able to see that this has allowed much more extensive processing, which has benefited biological applications fairly widely. It has also facilitated method development that we have seen been used and reused in other softwares as well, which has really been beneficial for the field in general. Reliant is an open source software, and it is beneficial that these methods get disseminated in any way or form. Moving to papers 3 and 4, they both consider a staple of, or historical staple of cryo-EM analysis, namely ribosomes. In this case, we utilized the chloroplast ribosome from spinach for 2 different purposes. Paper 4 investigates how fast we are now able to process data. Given the developments of paper 1, there was a capacity now to really accelerate processing and sample evaluation. And this was an investigation into the critical considerations for that. And what we found principally is that automation really is crucial for fast processing. The more you can automate data acquisition and processing, the more reliable the results are and the faster you can get them. Notably, however, there are a few points in this workflow that you will note are labeled in red. Those are steps which critically rely on manual intervention at the present time. So following any classification, we must select which data to discard and which to keep. And this is a critical step in most cryo-EM workflows that still is reliant on manual expertise or intervention. This prohibits automation and makes the results somewhat less objective. On a positive note, however, we do note that there are a number of additional tricks or measures you can take to speed up processing in terms of compressing data and processing small subsets. And it's still, based on those results, you can still extrapolate that to evaluate whether or not it's worthwhile to continue data collection to reach higher resolutions and so forth. So fast processing at a large number of cryo-EM facilities around the world is important to make sure that we make the best use of microscope time, which is limited, and it seems to be well-founded to do so. Paper 3 also examines the chloroplast ribosome, but in a more biological capacity. This structure displays some interesting properties that were not entirely expected. So the chloroplast ribosome is a specialized ribosome in chloroplasts that stems presumably from a endosymbiotic event of a bacterial ribosome at some point in the distant past. There are similarities between the chloroplast ribosome and bacterial ribosomes, but there are some notable differences. The structure that we were able to capture is the one you see in the hibernating state in the in the bottom left. So what we see there is that there is an additional protein that prohibits recycling by the RRF and additional recycling factors, and that stabilizes this ribosome in an inactive state. However, there are also notable distinct differences to the bacterial ribosomes, which are the presumed ancestor, that indicate that there might be a different method of nascent chain recognition by the SRP, perhaps involved in translocon recruitment. This brings me to the final project, which is the most extensive biological project that I will be covering. And this concerns the pyruvate dehydrogenase complex, which you also heard Sriram mention. We examined extensively the fungal PDC, so not the human PDC. And let me begin by saying that the PDC is constructed from 3 main enzyme components, E1, E2, and E3. If you processed any eukaryotic PDC, what you would expect to see is that the E2 unit forms a highly symmetrical stable core unit, and you would expect to see E1 and E3 be flexibly attached around its periphery. If you reconstructed the mammalian PDC, for instance, the human, you might then also hope to see, and this would be very difficult to see, but you might see that some of the core E2 units, this stable, rigid scaffold, is substituted for a 4th protein that specifically recruits E3. The fungal PDC is a bit special in that it has a protein that carries out exactly the same function. So it specifically recruits E3. However, it does not appear that this can substitute core E2, rather it binds to the PDC somehow. What we were able to show in paper 5 is exactly how this protein binds to the PDC and what limits its binding stoichiometry. Curiously, that binding stoichiometry is highly similar to the mammalian or human PDC as well. So it really looks like the fungal and mammalian PDCs have a similar function under entirely different structural rationales. On the left here, you see the PDC reconstructed from cryo-EM data we collected on the filamentous fungus Neurospora crassa. So what you can see is that we see the expected components, but we also see an interior component that seems to bind to the PDC core units. This is then presumably this additional protein, but it is not resolved that well, which we attributed to an over-symmetrization, i.e. it doesn't have the same symmetry as the core units. The key to resolving this was really to lower the symmetry to a tetrahedral symmetry, which is still compatible with the icosahedral core unit, but that affords a much better reconstruction. So on the right, we can now identify that the C-terminal domain of this extra protein called protein X in fungi, this trimerizes before it binds internal to the PDC core. By examining the size of this trimer, we can now also evaluate at which positions we can simultaneously bind such a trimer. And that enumerates a number of possible configurations for this interior component. Here you see a schematic scaffold on the left representing the PDC core. And on the right, you see a number of configurations where we have placed these protein X trimers according to what is possible in terms of steric restraints. So the top two, which are named X4S and Y4S, those are actually two tetrahedral configurations that are distinct from one another in terms of their reconstruction. So we initially only located X4S, that was the only one we saw, but by examining where we can place these PX trimers, we rationalized that there must be this Y4S state as well. Curiously, only by knowing that and extensively reprocessing the data were we eventually able to find that state. So unless we had done this, we would not have known that this existed. There are additional states that we can imagine as being saturated. This 2S state is, for instance, has these PX trimers located immediately opposite one another. It is in fact such that you cannot place any additional PX trimers in this structure. Similarly, you can place 3 in the 3S state, and you cannot place any additional PX trimers in that as well. An interesting question then is if these additional states also exist natively or in our data. And unfortunately, we are in the situation where we cannot fully discover the answer to that question because of how the reconstruction procedures are implemented. So because of the large symmetry mismatch between the interior component, in this case, in these configurations, and the icosahedral core, that is a very difficult challenge to tackle computationally. The issue really is that if we try to reconstruct this asymmetrically, the components that follow the symmetry might implicitly symmetrize it, or the prior belief on orientations formed by statistics regarding alignment might cause it to collapse as well. So the conclusion here is, unfortunately, that searching for lower symmetries and asymmetries is not really possible. And this extends to a wide range of biological application, not just this particular one, wherein a lot of biological assemblies form under symmetry. But it is enormously interesting to find deviations from that symmetry, however small. So there are a number of heteromeric biological assemblies that would be enormously interesting to tackle with such an analysis. And you might have multiple binding sites that you don't want to average together in a symmetric fashion, for instance. So another issue is that if we had a method that was able to isolate and sort of find these states, we could not actually trust any quantitative results coming out of such a classification. And I illustrate that by this example, wherein we take these two tetrahedral classes that I mentioned that we do find in our data set, and we take particles pertaining to each in equal proportion. So as you can see to the right here, we take 18,000 particle images of either class, and then we process that as if that was a homogeneous subset, i.e. under the assumption that there's only one state. What we see is principally not the mixture between the two states, but rather one overpowering the other. That indicates that a large proportion of the data here is being obscured by the reconstruction procedure. That's not principally an issue. This is part of the design of the reconstruction algorithm that it should be partially robust to inclusions of conflicting particles. However, it precludes us from making any quantitative statement, as if we didn't know any better, we might here conclude that there are 36,000 particles of the class that's reconstructed, whereas we know that that's not true. Again, this extends to a wide range of biological applications where we might want to infer quantitative knowledge from the classification output. However, drastic alterations do the reconstruction protocol would be necessary for that to happen. So again, this is highlighting a limitation of the method itself, but fortunately we can still draw some fairly interesting biological conclusions from our structure, examining the tetrahedral state where we see the protein X component well resolved. So here we see the binding site in Neurospora, so we can actually see where this protein binds to the core, and we can, by comparing it to the human and other eukaryotic PDCs, see that this binding site is entirely unique to fungi and ubiquitous in fungi, actually. So this seems to have occurred fairly early in the evolutionary branching between fungi and other eukaryotes. It is curious, however, how similar they are in function in their E3 recruitment and binding stoichiometry, but there seems to be, this indicates, if nothing else, that that's more due to convergent evolution than some sort of relation. So this precludes us from making any sort of inferences about the human PDC based on what we're seeing here, unfortunately. Nevertheless, to conclude this project, we really complement the evolutionary picture to some extent and see that there are fundamental similarities between the fungal and mammalian PDCs, but they are drastically different in how they are established. And curiously, this protein domain that we find has no prior sequence homology or known structure, so we can really now start to map a new protein fold, which is very interesting for me at least. And then drawing general conclusions about all of these projects and what I have been doing for the last 5 years is that being involved in development has been fantastic and very educational. And I have sort of been involved in and focused on speed and automation and a lot of qualitative improvements. And this really has been gainful for biology and the conclusions we can draw using cryo-EM data. But there are now, I would like to see this move into a direction where you can make more confident statements based on those outputs, and maybe perhaps have confidence measures attached to these outputs. I think more sophisticated descriptions of symmetry and asymmetry are needed. There are a number of fairly sophisticated ways to departure from symmetry, but this needs to be better understood, I feel. And so with that, I would just like to thank the people involved in these papers and the projects that I've been involved in. I would also like to thank the cryo-EM facility at SciLife Lab and everyone else involved for teaching me a lot of things over the last few years. And thank you all today for your kind attention.
Thank you, Björn. Okay, so we're going to stretch our legs for a couple of minutes. And also, I just want to remind the committee and opponent to indicate if there have been any problems in viewing what has been said so far. And if not, we will just take a short break. See you back here in 5 minutes.
There we go.
The sound on?
Yes. No.
Can you hear us? Okay. Very good. So welcome back, everybody. and we will now start the actual discussion. So and that is first led by the opponent. So please, Professor Subramanian.
Okay. Okay, let's get started. Hello, Björn. Good job. I enjoyed listening to you present your work. I understand that this, this essentially takes the form of a discussion. So perhaps we can begin with you identifying, now that you've spent 5 years working in this area, where do you think some of the major challenges are in single particle cryo-EM? You touched on some of those, but if you had to list, let's say, a few of the most important challenges, aside from the symmetry issue, which is kind of specialized, why don't you spend a few minutes perhaps outlining where you see the challenges in the field?
First, let me first ask, you can see me?
Yes.
Good. Yes. So there are a number of current developments that are fairly interesting in terms of cryo-EM, most notably moving towards lower energies. I think that's going to be an interesting challenge, and some of the challenges inherent to that, I guess, are already being met, namely different correction factors and things that would be required to make full use of lower energy microscopes, which would make everything much easier to process or much easier to collect, and it would increase the availability of cryo-EM in general. As far as single particle goes, it has up until now, and will continue for some time, I guess, to be highly purified samples that get examined. This is natural. So we tend to say that cryo-EM is so great because you can have a heterogeneous sample, but the truth being said, at the moment we have to really purify our samples as much as we can in order to have the best possible outcomes that can be interpreted with any reasonable degree of confidence. So what I hope we will be able to do is look at much more heterogeneous mixtures and better approach the cellular environment. One method to do that, of course, is, as you touched upon as well, this tomographical approaches where you look at the native cellular environment rather than opening the cell and trying to look at it with single particle. This is moving then single particle towards more of a subtomographic reconstruction regime where they sort of meet and you start to really try to classify the cellular environment rather than some purified sample. Is that sort of sufficient, or were you thinking of
Well, maybe we can drill down a bit more. You said that lower energies might make it easier to collect. In what way is that the case?
So it would really facilitate accessibility to electron microscopes. At the moment, there are a limited number of high-end microscopes around the world that can be used for high-resolution reconstructions. Moving towards lower energies would mean that those microscopes are cheaper. You can have more of them. It would be easier to screen more samples, and it would just facilitate more projects to be applicable to cryo-EM. That is what I was envisioning there.
But yeah, I guess, but when you say easier to collect, given a microscope, I don't really see why it's any easier to collect it on 100 kilovolt or 200 kilovolt than it is on a 300 kilovolt. You didn't mean that, I take it.
No. In principle, some of the issues that stem from using lower energies, as I mentioned, mean that you have to incur some additional aberrations that you need to correct for, and you can now do that to some degree computationally. The fact that you can do that sort of facilitates what has been coined sort of a shoot first and correct later, or align later. So you don't have to spend as much time carefully aligning your microscope in order to make full use of the data. That might be facilitated by those measures at lower energies as well. But in principle, no, it's not that much easier to collect data off of a low-end microscope or lower energy microscope.
And what kind of corrections do you think might be most relevant when you go to lower voltages? And perhaps, when you say lower voltages, what are you thinking of? What number?
So really, the lower the better in terms of how big the microscope needs to be, how expensive it is, and what would, sorry, I lost the question a bit there. What was the
energies? What voltage are you thinking of?
That's probably around 100 to 200 mark. Going down to 200 still requires quite a substantial microscope, and it does make it a fair bit cheaper, but there is promise of going all the way down to 100, where the information you can get per elastic event or per radiation damage is optimal, according to some recent publication. So I envision 100 kV being fully utilizable for standard investigations.
And what corrections do you think will be most relevant in going from 300 down to 100 kilovolts?
This is a bit of speculation from my side, because I don't know principally, but I'm given to understand that there are a number of optical considerations that, for instance, beam tilt gets more difficult to manage, and I don't recall at the moment if the beam coherence is affected, and you would have to incur some sort of correction factors for that as well. That I don't recall.
Yeah, beam tilt. I mean, I think beam tilt will be relevant. So will perhaps corrections for the Ewald sphere, which is mentioned in one of your papers that you're a co-author on.
Yes.
Do you have anything to comment on that? And if not, we can move on. I mean, I think that those.
No, in terms of if you go down in energy, obviously you have to use thinner samples in order to preserve the projection approximation, which you can sort of you can use Ewald sphere correction or some other correction to account for the fact that you may have a breakdown of the projection approximation. So I have a difficult time reconciling whether or not Ewald sphere correction is a straight shot correction that you can apply because your sample is too thick for low energies. I don't know if that's strictly true, because I don't see Ewald sphere correction taking care of, for instance, pleural scattering within the sample. I don't think that's possible. But in terms of the projection approximation, I think Ewald sphere correction could be useful in that lower energy range.
Mm-hmm. In terms of processing, that's the microscope. Still, at the end of the day, somebody has to make it. Somebody has to build it. Where do you see the gaps in processing going forward? You began to touch a little bit on this in the final part of your presentation. Maybe that's something we should look at, because clearly collection of the data is one thing, but facile processing is also equally relevant to making, solving some of these challenges. So can you expand a bit on that, in your perspective, on where the main gaps are technically for the image processing part?
So the most obvious gap, of course, is the selection of data, as I described in my talk as well. Are you referring to whether or not the processing would be undertaken in conjunction with data collection and sort of left out of the hands of the researcher? Is that what you're?
I think that's even further into the future. But given, as data is collected, no matter what the voltage, what are the biggest gaps going forward? I mean, you touched on one in particular in terms of classification and perhaps the population of projection images into classes that may not be completely appropriate. But is that the biggest challenge, or are there other challenges that may be more general that one has to deal with going forward?
In terms of the classification challenge that I mentioned, there's an obvious duality to this. So the first is that you would like to be able to identify different things in your data. Then there's a finer granularity that's more along the lines of what you described, where you might have conformational flexibility. Those are sort of overlapping cases, and they have different interests. And I think at the moment, most people have a much more direct interest in flexibility analysis, which there are tools being developed to do. You have different manifold embedding schemes that are trying to map the energy landscape of different flexible changes, for instance. And we know from before that dividing your data into distinct classes when you might expect a continuum of states is probably not the optimal way to do that. So in terms of flexibility analysis and classification in that setting, I think there will be fairly substantial developments in very, very short time.
To go back to, I mean, the flexibility piece, of course, is intimately tied to heterogeneity. So when you classify it, it's not obvious whether it's because of flexibility or some other reason. So to what extent does this business of incorrect assignment into classes, which I think you call marginalization, thesis, how relevant is that when you have no symmetry?
So how relevant is the effect of marginalization on classification when you have no symmetry?
Is it relevant? And if so, how much?
Yes, I would say it is. So in the thesis, I emphasize marginalization as having an effect on this, and that's because it's a very obvious. It exacerbates the issue quite obviously, because what it affords is an ability to take a particle image and rather than have it coherently contribute to the reconstruction, if it has an uncertain alignment, you spread it out so it has an incoherent contribution. That affords a degree of or a capacity to obscure that piece of data. So the reconstruction that you get out and the classification will not be, it doesn't make as strong a statement as you might think it does. So if you run a classification and you find 3 output classes, that's not a statement about your data saying that there are just 3 things. That's a statement saying that you have 3 things in your data, and this is one way of optimizing their 3-dimensional shape so that it agrees to in some sense optimally with the data. It would obviously be very, very good if there was a confidence measure or a heterogeneity measure of some sort that would make a claim saying like, these 3 things is what I found, but it doesn't completely agree with the data. There are more things to explain in this data. That's an extremely challenging thing to develop, and it's hard to envision exactly what it would look like. But I think that kind of statement would be necessary to get the most out of cryo-EM data.
Do you think that one could get it using variance maps, 3D variance maps that some of the early developments constantly use the idea of 3D variance? Do you think that'll help, or is this going beyond that?
I think it could be useful in finding things that you might not have otherwise found. I'm struggling to see how it would make a positive claim on any degree of heterogeneity. Of course, a variance map is to some extent an estimation of local signal to noise in the reconstruction, and that could be used to segment the data further. I tend to not favor approaches where you apply some form of heuristic to simply change the classification procedure. So you can imagine a classification wherein you would say, okay, so I have two reconstructions that look extremely similar. I want to merge them. Or I have some unexplained discrepancy in the signal to noise in the local region. I'm going to try to split this reconstruction into two different classes. That level of heuristic is of course possible, but it's hard to see how that would be rigorous as well. You're just tweaking the convergence behavior of the current algorithm as well. So you still wouldn't be able to make any positive claims based on the classification output. But it's certainly a tool that could be used.
So in the example you showed in your presentation, you mentioned the presence of these two, where you had two classes with similar numbers of projections that went into it, but you said, you pointed out how one of them actually dominated the average when you brought them all together. If I understood what you were saying correctly, is that right? That's your point is that one of them dominates more than you expect it to dominate. Is that correct?
Yes. So the reconstruction procedure essentially emphasizes part of the data, making the statement that this reconstruction exists within that data without really recognizing that there is something else that could be coherently contributing in a similar fashion.
But when you got these classes, you already had some symmetry, not icosahedral perhaps, but you had tetrahedral symmetry. Or was this done without any symmetry assignment? This is just straight classification with no imposition of symmetry. Is that right?
Yes. So we took the original data and applied tetrahedral symmetry because it is a subsymmetry of the icosahedral symmetry. So we knew that if we applied that symmetry and everything worked fine, we would still see the core assembly, but presumably we could also see something additionally. When we did that, we saw one of these tetrahedral classes. Then when we realized that there had to be another, we simply reran that analysis multiple times and eventually tweaking parameters and sort of setting to sort of affect the convergence behavior in subtle ways, knowing that we should see this other thing. And eventually then some of those runs were consistently outputting this other state as well. So it's a matter of luck, essentially, reprocessing until you find what you know must be there in this case.
How relevant is it to other examples you discussed? For example, the chloroplast ribosome, or maybe some of the other complexes you looked at. Is this kind of a small decimal point effect, or is this really significant in some of the conclusions you had where classification was indeed used extensively to look at multiple states?
I would say it's definitely, it generalizes to cases like the ribosome. I don't include any examples of this in my thesis because these relate to other projects, but I have seen cases where, for instance, you have extensive heterogeneity of composition in a ribosome preparation. In that case, there were many different classes to be discovered. But even then, isolating a single one of those classes and rerunning classification on that turned out to find subclasses within that. And it can be very deceiving. So one of the cases we saw a ribosome that appeared to be empty. It didn't have any tRNA in its binding site. But then following additional classification, the majority of the data turned out to have tRNA in there. It was just in varying conformational or invariant compositional states. So it's really remarkable how well data can be hidden to some extent. And this, I think everybody who has processed cryo-EM data knows that, for instance, during 2D classification, you would be surprised at what kind of data you can find that was never evident until later.
Yeah, I completely agree with you. I think that's very relevant, and I think it needs to be fixed. So I was curious what your thoughts were on how one might have general procedures to fix this, or at least to be cautionary about how to deal with the information that comes out of classification. I think recognizing the problem is, of course, the first step. Yeah. What do you think the solution is?
So we've discussed this I've discussed this with a number of colleagues over the years, and it's difficult to reconcile exactly how it would happen. It's a very challenging problem. One thing that came up recently was that one could do a level of meta-analysis to a certain extent. So if you imagine you take your input particle sets and you try to align them against the reference you have. Let's, for instance, say that you have particles that pertain to two completely distinct entities, but you have a single reconstruction that you're trying to align them against. Now, half of the particles then will not be consistently aligned, but there might still be patterns in the alignment weights that might reveal that there is a subset of the data that doesn't agree with the reconstruction, but that mutually behaves similarly. That level of analysis is something that could foreseeably be employed. I know there has been other people who have tried to look at migration analysis. So they look at how particles jump between different classes that you might want to reconstruct to try to infer that this doesn't fit anywhere. It might be something else, or it might be noises. But these meta-analysis that I've seen before have not been productive in the sense that omitting those particles, so trying to identify what these specially behaving particles are, has never yielded a better reconstruction or consistently reconstructed to something else. So it's difficult to say exactly what classification is classifying and what constitutes the differences in these assessments.
Do you think it'll go away if you collected much more data, say 100 times more data? Would it be easier to solve the problem?
Foreseeably, you could, if you have a very small subset of your data that is identifiable as something else. And it could be that you have one dominating state and then 90% of your data is 100 other different things. If you collected more data, foreseeably, each of those small populations could be sort of sufficient to reconstruct, and then you could identify them. But there are other issues that you would encounter, I guess, if you went to really large and very heterogeneous data sets. Notably, when you go to extremely large particle numbers, you start to actually come into numerical issues during your back projection and reconstruction procedures that you need to deal with. Currently, most of the software uses single precision because it's fast and reliable for the current task sizes that we use, but you might have to create special procedures to make sure that you don't miss things. It's really difficult to develop that feature though, because you would need to have a positive test case to see what's happening with very large data sets. And the problem with cryo-EM is that we don't strictly know what we have in our input data sets. So being positive in that statement during development is really difficult.
Do you have a feeling for what kind of numbers? What's the large number? Is it 1 million, 10 million? What are you thinking of? Where does this become a real problem? What size is this a real problem?
I haven't done the estimations myself, but I recall somebody making an estimation sort of back of the envelope that when you start to sum together millions of particles, at that point you might start to suffer. That being said, the way we do processing for large data sets is inherently distributed now, right? So you would actually reconstruct, if you had a particle data set that is 5 million particles, you wouldn't try to sum together 5 million images. What you would do is you would sum together maybe a couple of 100,000 in multiple locations and then merge them. And those reduction discrepancies and errors in reduction don't strictly apply in a simple way. But nonetheless, we haven't seen those things. I haven't seen them, I should say, but they might still be considerations.
Yeah, actually, this brings up the issue of speed, which you also spoke about, and of course, it's very interesting. Some of the work that you did clearly led to significant advances in the speed of processing. Can you touch on where the challenges lie in speeding up things? Is there room for further improvement? And if so, what does it take to get there?
There is absolutely room for improvement there. In terms of Reliant, that was very, I'm not going to say it was easy to get good speed up, but the algorithmic flow of Reliant is very suitable for parallelization. So it was very straightforward to analyze where we would need to go about doing things. In reducing the computational or the time taken to do these computations, the next trick, of course, is to make sure that you don't do all the unnecessary computations, because at the moment, Reliant really does a lot of work that it doesn't need to do, I feel. This is some of the success that, for instance, CryoSpark has had in that they very efficiently reduce the number of computations that you need to do. So in speeding it up further, I think it's not strictly as simple as making sure that you have better parallelism and so on, that like we did for this project. Rather, you would really try to cut corners that don't need to be computed.
Such as?
The orientational search is the obvious choice here. So there's a massive computational task in computing different alignments in Reliant, and there's really large regions of conformational space that don't need to get explored every single iteration. But at the moment they are, due to the rigor of the original Reliant implementation, where you do a global search over all possible orientations to a coarse degree, but still it is a global search every single iteration during classification, for instance.
Have you run benchmarks of CryoSpark versus Reliant? Let's say you had 100,000 particles. Have you done a careful analysis of where the gains might be in what CryoSpark does?
No, we don't. First of all, we don't use CryoSpark for anything. And second of all, it's fairly obvious from their implementation where the gains would lie. And I don't think we need to mimic their layouts or their method specifically. I think we know different things we can do to speed up Reliant in terms of improving the workflow and data workflow, and this sort of reduction of the orientational search. There's known methods to do this, and I don't think we need to sort of compare strictly. I should say, compared them one against the other to sort of try to estimate what kind of gains Reliant could see from doing things like they do.
But in terms of speeding up, staying with the Reliant framework, clearly there is quite a lot of room for improvement, as always. Where do you see that headed? Is it in terms of time reduction, in terms of maybe different ways of doing this? If you reduce the search space, for instance, that'll give you maybe what, a few-fold, two-fold, threefold? I mean, what order of magnitude are you thinking in terms of speed up you might achieve?
So it's really hard to be predictive about this because there's so many nuances to having a high performant algorithm, and especially when you're trying to schedule it on GPUs, because there's such a specific type of hardware that you need to tailor your algorithm to comply to. So for instance, if we would reduce the orientational search, which at the moment is global, that's very difficult to handle in an implementation because we no longer know exactly how much work there is to be done. So for the first particle, you might have 7 orientations that you need to examine, and for the next one, you might have 3,000, and then you might be back to 45. At the moment, the speedups we got from implementing this parallel algorithm is based on the fact that you know that you have, say, 50,000 things you need to do, and you know exactly how to schedule them for every single particle. That's the basis of the speedup, because Reliant does a ton of work, right? So the more you reduce the amount of work, the more complicated it becomes to sort of maintain that in a good way. But I still think that just avoiding it, you might see foreseeably somewhere between ballpark 2 to 10 times faster, depending on exactly how many corners you can cut. It really depends on the task size as well, which is enormously variant across cryo-EM. You can have very demanding alignments and classifications, and you can have things that just converge instantly, and you don't need to have that high sampling.
And would some of these problems be solved by just running it over more GPUs? I mean, it seems to me if the cost of GPUs comes down, could you achieve some of these things simply by having more processors?
To a certain extent, processing of larger data sets will always sort of benefit from scaling the resources in proportion. I think it's not really justifiable to just throw more watts at every single problem, not in the least if you consider sort of energy consumption, because I mean, we're living in a day and age where you need to actually be careful about your power consumption. You have to justify it somehow. And I think just throwing more power at a problem isn't really what we should be doing. We should try to be a bit more clever. And this comes back to if you reprocess your data 20 times in order to be confident about the output of a classification, you would rather have a method where you only need to process it once, and then it makes a statement about how certain you can be.
Yeah, I mean, that's a really good point. I think ideally you want to you want to minimize the amount of compute that has to be done. I completely agree with you that you want to be economical. But isn't it intrinsic to the way that you get these things, that the processing is done, that each time you do this, you will get a slightly different result? I mean, it's not, it's stochastic in that sense, right? Can you amplify on that? I mean, you still, it doesn't absolve you of having to do this multiple times, because there is a certain stochastic nature to this, the process by which we get to the 3D structures.
Yes, the current reconstruction procedures are locally convergent, and that convergence is stochastic, as you point out. So, and this is why we by reprocessing data, for instance, could find this other state. This is why we applied the approach that we can just run this over and over again with slightly different parameters, and eventually we might find the thing we're looking for. So that is probably perpetually something that you would want to do, but there's nothing prohibitive that says you can't do that by nonetheless implementing measures that might indicate to you whether or not that's a fruitful approach or not. We knew it had to be, but that's not always the case. You might have a sample where you think that you're seeing everything and you process it 20 times, and nonetheless you see the same thing, and that leads you to infer false things about the underlying data, right?
Yeah, I agree. I think that's a good approach to take. Maybe we can continue to sort of continue this extension on to tomography, which you touched on briefly. And I was curious to understand more about, what challenges you faced in applying tomography to get further insight into these complexes in the pyruvate dehydrogenase complex. Could you touch a bit on that?
Yeah, so originally the project I described in paper 5 was aimed at the flexible periphery, because this is fundamentally a very interesting challenge, and one that if we were, I remember saying at some point that if we could do this, then there's probably not much we couldn't do by flexibility analysis in cryo-EM, because it's such a vastly complicated problem. So initially we tried this with single particle, but then we realized that in order to deconvolute this flexible periphery and sort of isolate the information in a productive way, we had to do tomography. So we did that. I think a part of the reason for that project not being more successful is my ability to process tomography data, which is fairly complicated and not at all like processing single particle data. The challenges there, I think, are shared by many different projects, and trying to align and isolate extremely small things using tomography data is going to be a continuing challenge, and we're up against a physical limitation here, whereby the high resolution information is inherently attenuated in tomographical data. So while it's an interesting approach, it might hit a physical limitation that could be improved to some extent by, for instance, face plates, or if we see at lower energies that you can get better information retention. But as we discussed before, lower energies probably isn't the best approach for tomography, which typically has fairly thick samples as well. So I don't know strictly what the limitations are to processing things like the pyruvate dehydrogenase complex with tomography. But the attempts that I have made sort of indicate that it's right at the limit of what can be done.
But the samples that you looked at would have been sufficiently thin, I imagine. These were single particle specimens, so perhaps not as thick even as the work that John Briggs and co-workers have done with viruses. So at least from their work, it would appear that one could get to near atomic resolutions in tomography. So I was curious where you saw the differences between that kind of work and what you've done, keeping in mind that their structures were with entities that had some intrinsic order to them, such as the HIV gag lattice. Do you think that would have made a difference in your case, the fact that you had no symmetry? No order, rather.
Yeah, exactly. So the absence of order and symmetry is really means that we have extremely small things that we're trying to align. I don't recall exactly the tomographic or sub-tomographic averaging approach from the Briggs lab. I don't recall exactly what the size they were trying to align the sub-tomographic particle size. But I'm guessing it was to some degree larger than what I tried to align in this case.
I would say it's in the same range. You know, the lattice
Okay.
is slightly larger. It's, you know, maybe a few tens of nanometers, but similar, close enough to what we had. But I think I just want to get back to the use of tomography in your case would have actually been incisive in answering the question that you posed earlier on, which is, given the fact that you have classes, going back to the beginning of the discussion, where if you had classes populated by projections that shouldn't be there, ideally one would have thought that tomography would somehow reveal the answer. Is it fair that was is that your motivation in trying tomography?
So the motivation for trying tomography is really the fact that if you attempted to do single particle analysis of the flexible periphery of the PDC, the issue there, as you said, is disorder. So you have essentially non-identical particles. You can collect 1 million particles, but the probability that you will find even 2 that have all of their flexible peripheral enzymes in exactly the same conformation is very, very low. So you cannot coherently average that flexible periphery, which means that in order to quantify it to any degree, you need to isolate individual protein units in that periphery. The problem in doing so is that because you have multiple ones, you cannot, in a straightforward way, isolate the information pertaining to any one. If we could divide up the flexible periphery from a single particle into its constituent pieces, we could use it for single particle analysis and find out what we needed to know. Unfortunately, looking through multiple of these flexible components, we need to be able to align each one and subtract it before we can get to any other one. So it's sort of a chicken and egg problem in that sense. Conventional multi-body analysis that people do now is based on the fact that you can align all of the individual pieces, even if they are flexible with one another. That's strictly impossible for the PDC because of the large number of different and presumably independent flexible entities. So that's really why we moved to tomography, in order to deconvolve that flexible periphery to some extent and partition the information belonging to any one flexible enzyme. Mm-hmm. So this would allow us to sort of isolate the information pertaining to distinct flexible enzymes and maybe coherently average them using a subtomographic approach. So that was the motivation, really.
Yeah, I agree. That sounds very good. But also, if you cut out all of the moving parts, the core itself, you comment that you were surprised that the resolution didn't go to as high a value as you would have liked it to go to. What do you think the reason for that was? So
again, I sort of refer here to my shallow knowledge in tomography processing, because it might be due to choices in radiation dose that we use during acquisition. It could be down to an incorrect CTF correction. I know for a fact that we used a fairly rudimentary CTF correction, whereas I would like to have spent the time to do more sophisticated CTF corrections. All I know is that when I describe it to experts in the field, they say, "Oh, it should be better than this," which I agree with, but I don't principally know what the limiting factor is at the moment. unfortunately.
That's what experts are supposed to say. That is supposed to be. Yeah, okay. Now, I must say, I'm not too surprised myself because there are a number of approximations made in the business of back projection in tomography that have varying levels of problems to them. So at every level, there's some information that's lost. So ideally, you just prefer not to have to go through the route if you could just go with the projection images. But yeah, it's tough. I think it's a problem that is not obvious at this time. So I wouldn't be too hard on yourself because it's not. Yes, it should be better, but not too many people know how to make it better either. So I wouldn't feel too bad about it. Let's just go on to maybe continue on to cell biology. You spoke a little bit about where this is relevant to cell biology and to look at complexes in cells. What are your thoughts about that? I mean, how is this field, as you rightly point out, not everybody is going to want to produce perfectly purified samples to carry out single particle cryo-EM, but they want the answer. So how do you see the transition to cell biology, perhaps through tomography and other methods? And I was curious what your thoughts were.
Yeah, so I used to be of the opinion that you should strive to make everything as realistic as possible. And coming from a computational background, it's very easy to make statements about how close to native conditions you should strive to be in the lab. What I've discovered is that, of course, as most things in science, we isolate factors for a reason. So the purification and isolation of samples is strictly necessary for confident interpretation. So is there a middle ground really between, for instance, tomographical approaches where you look at the native cells cut open, or where you use purified samples? And I'm not sure whether or not you could find an intersection that would be very productive at the moment. Not only because the tools are not adequate to properly classify such enormous data sets that you would need, but just from a productive or a productivity standpoint, can you make confident conclusions if you try to extract the cellular environment? I think in terms of moving towards more native-like conditions, cryotomography is probably going to win out over single particle analysis.
And how do you see lower voltages help that cause? Probably not very much, you would think.
Probably not, but there will, as I mentioned, there will always be cases where you examine isolated samples for the value of them being isolated and sort of in a controlled environment. And in those cases, I think lower voltages will be useful. So I guess what I'm saying is that there will really be a bifurcation of those approaches. So single particle might not demand the current microscopes that we have, but I think that in terms of moving towards a more cellular perspective for cryo-EM, cryotomography is probably going to play a bigger role.
But let's say that you are looking at pyruvate dehydrogenase complex in a cellular context in Neurospora. Don't you think it's going to be a lot more difficult? I mean, it was hard enough with the isolated PDC. How is it going to be in cells?
Yeah, it's a good point. But I believe that if you wanted to study it in a cellular environment, the question perhaps wouldn't be what is the spatial relationship between flexible peripheral units, or what is the core internal protein structure. Then you would have questions to ask, such as where does it cluster? Does it interact in specific parts? Does it co-localize with other proteins that we can label? And in that sense, I think the cellular environment is a better place to start. But they're certainly addressing different questions. In this project, we are addressing very specific questions that benefit from that purification.
So yeah, I guess that's a fair point. But if you were to look in Neurospora mitochondria, I think you may not see very much, because don't you think that you will need contrast between the pyruvate dehydrogenase complex and something around it? If your protein density in mitochondria is a 50% or thereabouts, it's we're talking hundreds of milligrams per mil of protein concentration. How will you actually distinguish one protein from the other? That's not clear to me.
No, it's not clear to me either if it would be possible. Distinctly for protein assemblies that are fairly sparse, for the PDC core, for instance, it is hollow, so it's not a dense, compact thing that you would localize in an easy way. You can sort of see it fairly easily in single particle analysis, but as you say, in a cellular environment where you have a fair high level of other protein densities, it might not be a structure that you could identify very simply. But then I would also say that the intuitions here are that I can't see it, so it might not be identifiable, and that might not strictly be true. There's continuing development now for segmentation and projection matching to find these subcellular localizations. As we go deeper and deeper into cryotomography and cellular environments and so on, these tools need to be developed, right? And I think that I think it might be surprising how well computational tools are able to find these things, even if we anticipate that they might be difficult to observe or localize.
Right. That's a good hope. But I mean, I think at the end of the day, you pointed out how it's difficult to be sure about classification, even when you have really good contrast. I think that some of the challenges in being sure that you actually can find a complex in mitochondria of a cell, I think that's still some distance away, because it's you have to have contrast, right? Otherwise, you will not be sure about what's being imaged.
No, I'm not sure whether or not tomography of a cellular environment would help answer the question of which, for instance, which internal configurations of the fungal PDC exist. Even though we might want to look at that in a native environment to ensure that the conclusions we draw are reasonable, I'm not sure you could do that. No, I think that's a granularity where I would also agree that classification of the cellular environment might be a challenge on that level, yes.
But in this particular example, if there was one thing you would want to know from looking at it in situ, what would it be for pyruvate dehydrogenase? What could you learn that you haven't learned already from looking at it in isolated form?
That's a fairly open-ended question. It sort of depends on what there is to be known about it, right, which we don't know. But certainly there are additional interacting partners that could be considered. There's a number of different phosphatases and kinases that interact with the flexible periphery as well. Whether or not it localizes to certain regions of the mitochondria might be an interesting perspective in that sense. But structurally, I don't know that there are any questions that I would tackle with the tomographical approach and in situ imaging for this specific case.
Yeah. Okay. No, fair enough. I think it's hard to anticipate, and I suspect it'll depend a great deal on the certainty with which you can identify PDC in a mitochondrion versus something else that's in the mitochondrion, and I think that the solution is not really clear to problems like that. Great, I think.
Yeah. Sorry, I was just going to say that you might also imagine that there are a great many transient interactions within cells that we don't know about because they are weak enough that we don't see them in our purified samples. So that's ubiquitously the case throughout biology, right?
So in the work that, for example, the Baumeister group has published a lot on, why do you think they can see proteasomes while it's not so clear you can see so many other kinds of proteins? So what is it that makes it possible to see proteasomes, you think?
I'm not sure if they are particularly dense objects. They're fairly large, which makes that a lot easier. But compared to the PDC, for instance, it is a much more dense structure, I would imagine. Then there might be different modification or post-translational modifications to it that might make it more visible. Or so typically, phosphorus or phosphate groups tend to light up in cryo-EM, as well as if you have cysteine bridges and cysteines in general, things like this. I'm not too familiar with proteasomes so that I can speculate wildly why proteasomes specifically are easy to observe. But I know which images you refer to, and they can quite remarkably find and match and localize them. So I don't know what facilitates that specifically.
It's hard to tell, but I think that it's not so much the modifications, but so much the fact that if you have a relatively dilute cytoplasm, it stands out as a larger protein among others. So I think it really boils down to having differential contrast of the entity of interest, whether it's a proteasome or a hexameric enzyme, which they've studied recently. I think it's still, whatever it is, has to stand out against the background. And I think methods that can make that happen, combined with computational advances, clearly will be important for the future. I just want one or two minor questions to finish up. I mean, if you had to combine all the work that you've done, say single particle cryo-EM and tomography, is there, can you name a interesting biological problem that you might apply all of this to that has not been looked at so far? Is there something that you have a burning desire that you would say, well, I wish we could have studied problem X with all these wonderful methods? Any thoughts?
Certainly. When we started this method development, we were fairly naive in what we assumed that this could be applied to. So from then on forward, it's really been of an interest to try to look at what we've been talking about, sort of as native conditions as possible. One thing that I would love to apply this to is different transient interactions between cells. So cell-cell interactions and signaling complexes. I think these would be approachable both by a single particle in isolated form, and I think cryotomography would be very gainful to gain perspective on exactly how different components of cells co-localize and localize to different extents. So combining those approaches, I think, would be very useful for a vast array of different biological inquiry.
Yeah, great. What was the hardest thing you did in your thesis?
Track down extremely elusive bugs, but luckily I had a great colleague that was able to be much more successful in locating them. Beyond that, I think going back into the lab and moving back from the computational aspect to the biological aspect and really grasping all of the technical things and practical aspects that you need to master in order for such a project to be successful. That was also very daunting, but at the same time, extremely enjoyable.
Great. Thank you very much. That was very good to hear your perspective on these things.
Okay, thank you very much. So we will move on to the committee. Do you want to start, Suparna? Just happened to see you on screen. We can't hear you.
Yeah, I think now you can hear, right?
Yes.
Yeah. I must say that this is one of the best defense I have attended in recent times. Not just because of the presentation, but the discussion was really high level. And I think as the credit goes to your opponent, Dr. Subramaniam, it goes to you also, Björn. So it's a great job and I learned a lot. So I start by asking you a question that I was expecting that will be touched, but it's not touched very much. As you know that I am an end user of cryo-EM. I solve ribosome structures using cryo-EM with different components bound on it and for different bacteria or other organisms. So we all know that in cryo-EM field, there is a huge paradigm shift that was touched upon, that before that it was like in Sriram's slide, like the blue blobology part, which now migrated to the golden era with high resolution. So can you explain what actually caused this paradigm shift in cryo-EM field?
There's a number of factors that go into this, and I always struggle to remember all of them, but fundamentally there was simultaneous technical developments that allowed much better information retention from the microscopes themselves. So the images basically improved. The digital readouts also allowed us to sort of correct for the motions that occurred during acquisition. So there was a bunch of method development there that coincided with that and complemented it, that just produced so much better input data. Then you could also argue that in conjunction with that, the software implementations that had been around really started to develop and use new methods in order to make better reconstructions. That's sort of a hard point to push though, because the cryo-EM reconstruction, as Sriram pointed out, has been around for a long time, and it's been able to do these things. I think the main limitation is actually technological. So the detectors improved, and our ability to correct and retain image information based on those technological developments really is the key.
Yeah, so I think, so the last point that you say that the detector has been improved. So for us, the end users, this is what we know for sure, that the detector has been improved. And can you elaborate a little more that what was the drastic change in the detection method that led to this paradigm shift? Yeah.
So there's two aspects to this. As I mentioned, the fast readouts is really crucial. So when you collected before, you collected on photographic film. You can't really change that photographic film every 1/10 of a second or so. So really be able to have digital readout fast enough to segment an image into frames allows to correct for motion, and that's fairly crucial. Then there's also the fact that there was a type of detector, which I assume is the one you're referring to, the direct electron detector, which was able to localize where the electron impinged on the detector to a much greater extent. So basically, we were much better able to localize the electrons hitting the detector.
Yeah.
The combination of those is actually another factor in the sense that we could do counting mode acquisition. Again, the fast readout means that we can now sort of detect a single electron, so we can be sure that when we see a detection event, that's a single electron, and then we can localize it even better. So really these things work together.
Okay, I'm satisfied with that. So I can ask you something, a very naive question, again, as an end user of cryo-EM, that because you really have discussed a lot about the heterogeneity issue and classification. So for example, if we take your example, if you work on this chloroplast ribosomes, so in the first paper, that 24-hour structure determination, that paper was focusing mostly where you could do the speed up so that it could be so fast structure determination. And then the actual structure was published where we see the ribosome, the chloroplast ribosome recycling factor RRF, and the hibernating factor, which is PSRP1 on the ribosome. Okay. So I have actually 2 or 3 questions which you can, if you explain, then I think that it will be good for everyone. So first of all, that when you did this initial very fast structure solution, did you see these extra densities for RRF and the hibernating factor or not?
The chronologically they are sort of opposite. So a lot of biochemical work was made to establish the purification and biochemical protocol that led to the sample preparation. That was then simplified to do this examination of fast processing. So to that extent, we basically took the resolution estimates from the fast processing at face value and didn't do a very in-depth analysis of the structure in that case. So what I know is that it was essentially similar. There was no surprises because it would be stupid not to check whether or not something else manifested in that reconstruction. But as I recall it, there were no substantial differences. So I expect that that contained the RRF, which I think the only time that RRF was not seen was when the sample was not chilled during the preparation to the same extent. So I wouldn't go so far as to infer anything about when and how that RRF produces a hibernating state in our samples. But I don't think that there was any extensive analysis to quantify the differences between the structures determined with the fast or rigorous method.
The reason I say that because when I read the materials and methods for your both papers, the ribosome prep is written identical, and there is no mention of adding RRF to it or the hibernating factor. So I assume that these were bound to ribosomes. And when I read actually how the particles were processed or classified, I do not see any mention that how many particles were with this RRF, how many ribosomes were with this RRF and the hibernating factor, and what fraction of the ribosomes are without it. So do you have any ideas about, or it was like purely homogeneous sample?
Well, in line with my argumentation around classification, the easy answer to that question is that we couldn't know, even if we classified, even if the classification said that 70% of your data has an RRF and 30% does not. That would only be a statement saying that you have some with RRF and you have some without. Because again, classification is not quantitative, right? So, but that's a bit of an escape. I think in this case specifically, there was no dominant sample that didn't have the RRF. So basically it was a complete hibernation of the sample, apart from perhaps contaminating large subunits in isolation. And I don't recall if we saw any cytosolic ribosomes in this prep actually.
I expected, so I expected very high occupancy of RRF and the hibernation factor, looking at their density maps. So I expected that it would be present in most of the particles actually. So leaving that, I actually have one question, which is a bit more speculative and philosophical, that now that we see also from Dr. Subramaniam's presentation that cryo-EM is going towards structure solution of more and more smaller proteins. So do you think that this cryo-electron microscopy can really take over whole X-ray crystallography field, or it will abolish the need of X-ray crystallography?
It's very easy to answer that question. It's like, no, I don't think cryo-EM has any business trying to take over X-ray crystallography. I think they will find different niches in the present case example. So if you look at this internal components of the PDC that we have established, the most tractable approach to get an atomic level or an atomic resolution of that protein folder to better understand what it looks like would be to overexpress that protein for crystallography. That's a very valid approach, and it's much better than trying to squeeze this out of cryo-EM. Looking at smaller particles is always a better thing. I think cryo-EM should strive towards that end, because you might always have structural flexibility that cryo-EM will be more suited to deal with than, for instance, crystallography. But I think integrative approaches in biology is really the way forward. Computational methods sort of unify these approaches, and hopefully we can find sort of methods and invent methods that utilize data from both in an integrative capacity to sort of explain those things. Yeah.
Okay, thank you. I think I'm satisfied.
Okay, thank you very much. Let's see, Ozan, do you want to continue?
Yes, please. Thanks, Björn, for a nice presentation so far, and also thank Shiram for a nice work in surveying the field. Well, being a mathematician, I'm going to be, I'm on the other end of the spectrum, not on the user end, but perhaps more on the foundational side. So I'm going to maybe ask a couple of questions that are a bit unusual, perhaps. So let me start. You have an inverse problem, right? So that is a reconstruction problem, and it has a lot of nuisance parameters, which you call the alignment, which is orientation and in-plane translations. And then you also, on top of that, you have the, in the case of heterogeneity, and if you assume discrete heterogeneity, you have these classes, right? The structural classes. I'm a bit kind of curious, because when you're dealing with an inverse problem, even the tomography problem by itself is ill-posed. So you need to do regularization, right? You need to do something to stabilize the recovery procedure. And that kind of amounts to accounting for prior knowledge. And in your thesis, in the survey part, in the introduction part, not in the papers, you name drop a bunch of methods. And then, but most of your work has been with RelyOn, right? So I'm going to a bit focus on that. What kind of prior knowledge are you guys really using for the entire problem, which involves recovering the classes and the structure. And what does it translate to in the biological sense?
What biological prior information we use is effectively none, as far as I know. We employ several priors on that we partially estimate them from the data themselves, but we also have some assumptions of smoothness that go into prior belief to regularize the reconstruction problem. Is that what you're referring to in terms of the biological regularization?
Skip the biology part and let's keep mathematical. So you talk about smoothness, right?
Yes.
So and if I read in the rely-on papers, because I mean you haven't really modified rely-on approach, right? I mean you actually speed it up. You've been taking part in elevating the method, but really not really touched on the basis of it, right?
No, we have not changed the methods of regularization or reconstruction.
Okay. Yeah, so I cannot hold you responsible for the choices, but I can nevertheless ask you.
well, do. That's fine.
Yeah. So you guys are using, well, yeah, in some sense, Shores and companies using a Tikhonov type of weighting in the frequency space. And then there's a claim that this somehow corresponds to smoothness. Now, when I think about it, smoothness translates to decay of Fourier transform, right? That's the mathematical theorem. If the Fourier transform decays fast at infinity in any direction, then the function is smooth, right? And if it doesn't, then you have less degree of smoothness. So how do you how does this translate to this energy function? I don't get it.
How does this translate to the
choice of energy function? Why would that give you smoothness?
So I've understood this slowly over the last 5 years, how we applied this prior to enforce smoothness. And the first time I read the rely-on papers, I didn't actually understand it either. I'm still not sure I completely understand it, but the way I think of it now is that you enforce smoothness by basically truncating the spectral contribution to the reconstruction. So effectively, you're low passing it, right? So you're removing the higher frequency components in order to not have an artificial amplification of high resolution components that give you spurious details.
But in that case, why not just do an MLEM with a low pass filtering at each step? Why bother with energy functionals and stuff like that? I don't get it.
So at the moment, this prior knowledge that sort of enforces smoothness and regularizes the reconstruction has some mathematical or theoretical ground in that it's estimated. It's a parameter estimated by maximum likelihood from the spectral composition of the reconstruction, right? So it's to some extent based on the reconstructed quality rather than some arbitrary low-pass filtering. But perhaps you were referring to a low-pass filtering that is not arbitrary, but estimated somehow differently.
So you mean that the prior is updated in each map EM iterate? Is that what you mean?
Yes.
Okay, so you're updating your prior from the data along the iteration. So you're changing the regularization at every step.
Yes.
Okay. And this updating parameter, which we would typically call the regularization parameter, is computed by some kind of a maximum likelihood estimation from a reconstruction list, I guess, because, right, it cannot be from data, it must be from reconstruction.
No, so I include an expression in the thesis that describes exactly the update rule for this parameter, which would be equation 3.3.
Is it the one that you split the data in two halves and then do the Fourier share correlation computation?
No, not specifically. The one I refer to is on page 31 of my thesis and details an MLE estimate of the regularization parameter as evaluated from the reconstruction. So it's basically the spectral average of the reconstruction.
Okay.
That's what's utilized in classification. As you point out, when you do the refinement, you estimate the signal to noise ratio, and from this you can then recover this this variance parameter of the spectral composition and use that instead, which is a bit more rigorous and part of why refinement is more rigorous.
Oh, okay, you called it rigorous. Okay. Let's go on. So you are nevertheless a computational guy, right? So if I would say, I'm not really happy with that, with this energy function, I want to try something else. I want to try a regularizer, which is an edge preserver, you know, something like this, a bit different than a smoothing thing, right? Nevertheless, when you're looking at molecular structures, your predominant, I guess, edges are important since they give the overall shape of the object, right? So if I would swap that prior to something else, which is not a squared Tikhonov style in the frequency space, I mean, let's say an L1 type of total variation type of energy, what computational consequences would you expect from that?
That's a really difficult question. I don't know specifically about using an L1 regularization. What effect that would have, I cannot speculate on. But let's say that I use a different regularization technique, and what might I expect to see happen differently? If you then under-regularize the problem, then I could see that you would inflate your high resolution estimate. So you would get spurious signal, and you would basically introduce noise and false artifacts into your reconstruction. On the other hand, if you over-regularize the problem, then you would smoothen it too much and you would lose high resolution detail. And that's not something you want to do either. So it's a fine line you're treading, right? So exactly what regularization is optimal.
You misunderstood my question. Let me try to rephrase it. I mean, what you're saying is valid for any regularization method. So let me rephrase my question. So I would say there's actually a particular advantage purely computationally. Disregard, I mean, every choice of regularization could be bad or good depending on your real prior out there, right? Which you don't know. But apart from that, there is a specific advantage of using this quadratic type of regularizer, which is purely computational. Okay, it has nothing to do with regularization properties by itself. It's a purely computational advantage in the map-EM scheme that is used in Relion.
And are you then referring to the functional form and the fact that it's useful in the reconstruction or back projection estimation, that it forms this sort of Wiener filtering property? Is that the?
Well, actually, okay. Let me put the answer there. Essentially, when you have a quadratic data loss, which you have because you're also using the L2 in the frequency space in the data space, and you have a quadratic energy functional, you can actually compute one of the two steps in the map. I mean, if you write map-EM schematically, you'll have two steps. You have the E step and the M step. E step involves a computation of expectation, and the M step involves a maximization computation, where you're actually supposed to solve an optimization problem in the M step. So when you have a quadratic functional as a regularizer and a quadratic data loss term, you can compute the M step analytically.
Yeah.
You don't have to solve an optimization problem, which of course has dramatic implications on the computational feasibility.
This is a different way of saying that you can formulate the back projection operation as an optimal reconstruction method.
No, I wouldn't say that. That's something else. I mean, back projection is just that's the derivative of your forward operator, right? That's just the adjoint of the derivative of your forward operator just happens to be the back projection in your special case. No, what I mean is that the MLEM scheme, even if you take a more advanced, let's say, image formation operator, which is not a projection, it could be some wave propagation or something like that, right? Even play with the idea of using some multi-slice thing, some nasty nonlinear beasts.
Right.
So you won't have a back projection anymore. You would have some kind of a back propagation thing, right? But despite that, the MLEM scheme, just because you choose a quadratic regularizer and just because you happen to go with a quadratic data loss term in the frequency spaces, allows you that you can compute at least the M step analytically, which I would assume is good computationally, right? You don't want to solve an optimization problem at each M step, I guess.
Yeah.
But out of curiosity, then, I mean, when you guys recoded parts of Relion, So you recoded also the projection code, the forward projection code?
The forward projection code is one of the main components that needed to be accelerated, yes.
Yes. So you coded that on the GPU. Now I kind of bounced up a bit because I saw that you guys are computing the forward projection for each. I mean, you're boiling it down to pixel level almost, right? And that's at least the impression that you give in the text.
Yep. No, that's correct.
So why would you do it that way? I mean, why would you, mathematically, why do you want to consider the footprint of each pixel as a random variable? It's a bit odd. And why would they be independent? I mean, the voxels are not independent.
So are you referring to the methodological choice of doing that, or the implementation choice in what is faster?
I'm right now at the methodological part, not at the implementation part. I'm just curious, why would you assume that, why would you even need the assumption that the footprint of each pixels, footprint of each voxel, single voxel is supposedly independent? I don't get that.
So the methodological construction around the likelihood function sort of necessitates the assumption that they are all independent in order to compute this sort of distance metric that we use to compute the likelihood of each alignment, right? So
not at the voxel level, though. I have to disagree. I mean, I would say it makes sense to say that a random molecule, if you do a random draw from nature, you would expect them to be independent, right? But if you have a molecule at your hand, and then you look at let's say, okay, let's compute the electrostatic potential that this molecule is generating in this aqueous environment, and then you go into a specific voxel value, I wouldn't expect that voxel value to be independent from the neighboring voxel value.
No, and we know that this assumption is to some extent false. So we are doing. You don't need to make that assumption in order to be able to compute the correct quantity. I agree. We employ it because it makes a straightforward algorithm that behaves very predictably and is fast enough. When you make that assumption, you to some extent neglect correlation between pixels. Now that could be used, and it could be how do I put this in a good way? We compute the contribution from each pixel, assuming that they are all independent, while we know they are not. If we accounted for the fact that they were somehow correlated, we could presumably omit a lot of computations accounting for that. But then we would have to consider how they are correlated and how that influences the alignment metric, for instance, the likelihood calculation. And I don't think you could, of course, try to do this, but it's not something that we did. I will say so much, and I don't see immediately how we would go about doing it, although I don't doubt it could be done.
Okay. Going over to the margin, did you guys also elevate the marginalization code that shorts that? Did you also modify that one?
The marginalization code?
Yeah, the expectation over the orientations. Did you also modify that part?
Yeah.
Okay. Just out of curiosity, so this expectation is computed at during refinement, for example, it's computed at each iterate, right? Over each particle. So what is when you're creeping down to your atomic resolutions? Let's say everything goes nice and your refinement seems to converge, and you're crossing your fingers and you're hoping that you're going to get almost atomic resolution, right? So how finely do you need to resolve the orientation space?
So there's a fairly straightforward calculation that you can do based on trigonometry to just evaluate at what angular sampling density you need to be for contributions at a given resolution to contribute. Because if we're doing this in Fourier space, which we are, right, then you can see how if you modify an alignment parameters by a certain degree, then you rotate the central slice in the Fourier volume, and that means that higher resolution components move further. So that's a fairly sort of primitive way of looking at it, you could also, of course, account for an expectation of how coarse the alignment space is at that resolution. So if you expect high resolution components to change a lot in that resolution range, you might need a finer granularity, but I don't think we employ that at the moment. I think it's a straight estimation saying that, okay, so if we rotate it by this much, then this level of resolution could foreseeably have changed.
But if I say 3 angstroms, let's say you want to go down to something like reliably resolving structures of size of 3 angstrom or something like that. Are we talking about a quadrature that needs about 1,500 points on the sphere, or is it like 2,000? What are we talking about?
In ballpark numbers, yes, around I think for 3 angstrom resolution, then typically it also depends, of course, on the particle size and the box size. But I would anticipate several thousands of orientations need to be examined, yes, if not more. Typically, however, when we reach such high resolutions, we've already progressed to a local optimization scheme where we don't do a global investigation of all possible orientations, because at that point, we're fairly satisfied that the alignments are pretty well centered, and then the sampling density is automatically increased around the previously found optimum.
Okay, so the idea is that somehow that during refinement, the orientations don't jump around wildly, but
yeah, this can be a problem. This can be a problem if the resolution is drastically overestimated, because at that point you start to assume things that are not true about your data, and then you can get reinforcement of noise and just straight up collapse. And this tends to be very prevalent in highly symmetrical reconstructions as well, because the signal to noise there is elevated by symmetric averaging.
Okay. Then you had a discussion also with Shiram about going down to the lower energies. The argument was that you might get away with some cheaper microscopes in that case, but I thought a lot of the money was into the pipeline, right? Keep preserving cryo conditions and things like that, not necessarily the lenses and the magnetic field. But I don't know. But I'm just really curious. I mean, when you go down to lower energies, don't you have more impact from, I mean, if you disregard the lenses and optics and just focus on, let's say, the specimen electron interaction. I mean, doesn't it become harder to, even though your specimen is thin so that you get enough that goes through, wouldn't you have more, I mean, given the coherence length of the electrons, wouldn't you have more inelastic interaction when you go down in energy?
Yes, presumably you would have more inelastic interactions. I think the basis for the current developments towards lower energies is an article that was published wherein they found that the amount of information you can extract per inelastic event, so basically how much information do you do and how much damage do you do, that balance is sort of optimal around 100 keV for thin samples that are thin enough. If you have thicker samples, then as you say, I mean, you couldn't hope to get away with lower and lower energies, but there seems to be some sort of optimum that's compatible with conventional single particle analysis around 100.
Well, okay, Björn, but that sounds like a mathematical existence theorem, because one thing is that there is the information there, and that's probably true. The second question is extracting that information. That inverse problem becomes much more nastier, because I guess you lose coherence, right? You don't have that nice phase contrast type of interference thing. And your specimen is changing quantum state. And yeah, I just foresee in front of me all kinds of how nasty the inverse problem becomes. So I mean, do you have any ideas what kind of form model you would need to move to if you go down to 100, let's say?
I mean, I think it's desirable to move to a more sophisticated forward model anyway, but I don't think it's justifiable at the energies we typically use now. But as you say, if we move down in energies, you might have to. Nonetheless, I think if you look at it's one of these curiosities of cryo-EM that you can do single particle analysis on, for instance, viruses that are a micrometer thick. And if you do the back of the envelope calculations, they should go to something like 5 angstroms, and then incredibly you get 2.8. How does that go about? And so I agree with you, the inverse problem is nasty, but it seems to overcome a lot of the issues. I'm not saying that we should rely on things to sort of solve themselves in that capacity. But there are certainly aspects of the processing that seem to favor being able to do things in spite of these approximations being fairly violated.
So,
I don't
any idea
know.
why?
No, I've been trying to reconcile this. This is one of the things that you slowly learn over 5 years, and then you still don't know exactly what you know. I think in terms of, for instance, the thickness of viral particles that are that big that still reconstruct to a better resolution, I think there's some arguments to the fact that we apply a singular CTF correction that applies, that is constructive for part of the structure. The parts of the structure that don't adhere to that CTF correction, for instance, they are incoherently averaged or constitute a noise component that we can average away anyway. Essentially, we have algorithms that are really good at extracting the mutual information and disregarding that which is not coherent with that picture. That's as best as I can rationalize why things work when they shouldn't.
Okay. Well, I mean, of course, I think part of the answer also lies in what you mean by the notion of resolution here. And then at the end of the day, when people interpret their structures, they might also have a lot of prior knowledge about what they're looking at. I mean, it's not that it's totally blind. It's just like this experiment where you take a random person from the street and put them in a CT scanner, and then you give the image to the radiologist, and you don't say anything. You just silent, and then ask the radiologist to give an assessment. That's very, very different from a situation where there is a referral from a doctor and so on. I might guess that a lot of prior knowledge plays in the background here also when we say that we reach certain, let's say, information content. But that's just my personal guess. But it's an interesting question. Why do we somehow seem to breach seemingly limitations from noise and other aspects? On the other hand, I mean, you've done a lot of, you seem to be involved in a lot of, high performance computing things, right? And I'm assuming when you're recoding the stuff that was done in original Relion, you guys were using an infrastructure. I mean, you are probably a good programmer, but I don't think you rebuilt everything from ground up. I'm assuming you are using existing tools. So I'm a bit curious about if that's the case, of course, if that's the case, how generic are these for other tomographic problems? I mean, how transferable is the computational tool sets that you guys have versus how hard tied is it to particularly wave imaging or even phase contrast imaging, or even so cryo-EM imaging?
So if I understood the question correctly, you're asking how transferable are the implementations that we've done to related fields and problems?
Yes, like for example, your projection code. If I want to say, I want to use this for radar imaging, can I do that? I mean, how well encapsulated and abstract, how well API is, and how well structured is the code?
I think there's development in the cryo-EM field towards more of an API setting for the kind of tools that we did. We actually tailored it highly specifically for the task at hand. And that was because the first approximation, we foresaw a need for fast processing, and Relion was very extensively used. So we could have broad impact with it as it was. Modularizing the code and making sure that those functionalities are more generally applicable was never a priority for us at that point. It's starting to become one now, but there's an ongoing discussion on and different opinions in the field about how much freedom you should leave to users to customize their own workflow with that kind of modularity. So at the moment, Relion takes a very sort of protective or a very protective view, saying, okay, so there's fixed pipelines and there's fixed regularizers. You can tweak this in a bit, but mostly this is handled internally so that you can't make incorrect decisions. If you increase the modularity, then you open up for the potential, I'm not going to say misuse, but incorrect use of those functionalities. So there is some discussion and opinions regarding how much of that should be left to users. As you say, there is a lot of prior knowledge that goes into assessments in cryo-EM at the moment, and the evaluation procedure is not always entirely objective.
Okay. Well, that's not specific for cryo-EM. I would say even medical imaging is you might measure PSNR and SSIM, and so at the end of the day, it's a radiologist that looks at the images and makes the assessment. So it's not really surprising. So it's a nice piece of work you guys have been doing. I think I have one final question then, and I'll let you. You guys talked a bit also about heterogeneity, and you mentioned that this, I forgot the name of this object that you were looking at, or the molecule, but you said that it's one of the harder ones because it's so, I can take millions of these almost, and I will never find two examples that are exactly structurally identical, even though they come from the same molecule. So this kind of indicates that it's perhaps more reasonable to ditch the discrete heterogeneity assumption and go for some continuous heterogeneity, that you would assume that there's a deformation operator that acts on some kind of a template, and then you need to parameterize this deformation operator, and then that will be part of your inverse problem to recover those parameters. Is that anything you have been doing along these lines or something like that?
So there is a natural approach to employ in that case. The multi-body approach, which you may or may not be aware of, basically makes the assumptions that parts of your structure are rigid, and then they have some level of flexibility with respect to one another. And these are the parameters that you refer to. So in the case of this complex, what you would then have to specify is around 61 different bodies at least. And then you would say that, okay, so each of these bodies has presumably then 5 degrees of mobility. So you would end up with something like 300 different degrees of freedom in each particle. So I don't anticipate that would be productive, because you would run into overfitting issues way before you have any significance of fitting. And that's really supported to some extent by the tomography approach we did, where we try to isolate just one of these bodies and try to align it. And even that is difficult. So as soon as you allow conformational freedom to these small bodies around this enzyme, it tends to be very difficult to get significance. And parameterizing the motions of each one for each particle is I think, too daunting a challenge to attempt with single particle.
Okay. I think I'm done.
Okay, thank you very much, Hassan. Shadon.
Yes. Yeah. Can you hear me?
Yes.
So thank you, Bill. That it was a very nice thesis I read, and you know that I'm an electron microscopist and I'm a material scientist. So I'm interested in the method. So I will first ask you one question about the general application of this single particle analysis. As the opponent already discussed about, the development of this technique has been very so great, so that basically you can study larger molecules and smaller molecules. And I want to know if you keep your imagination, so that what is the limit of the method? For example, in terms of the size of the particle versus the resolution of the structure reconstruction.
There's a ton of additional factors that unfortunately play into this. So, for instance, if you look at a small particle, that could be rich in contrast, depending on what it's composed of. I think there's a fundamental limit that people are very interested in knowing, depending on how small your particle is, how well can it be aligned. So that's basically, if you can't align your particles, then there's no hope of recovering high resolution information. I have a basic sense of what I believe is possible, but I don't have any quantifiable evidence for these things. But certainly small enough proteins like hemoglobin and so have been processed to I believe sub 3 angstrom resolution in favorable cases. Whether or not that was under faceplate or if the faceplates even helps for these kinds of things, I don't recall exactly. In terms of larger structures and how well you can structure, how well you can resolve larger structures, that's entirely dependent on how flexible they are. Again, these large viruses should be more flexible and present a bigger challenge to high resolution reconstruction than they do. And it's really difficult for me to speculate on exactly what kind of resolution you can reach there. But certainly larger complexes have some inherent flexibility and bending modulus essentially. So single particle averaging needs to be able to account for flexibility for those cases.
So assuming the flexibility is not a problem, so that so are you able to so see that if you can get like 1 angstrom resolution, like in comparison with X-ray diffraction, what would be the computational requirement, or do you have a limitation of the processing or the in terms of the method itself, in terms of computation?
Do I understand you correct that you're asking what the computational challenge, the computational?
Yeah. So if you have a very large molecule, you have to have a lot of pixels at the same time. You have to have a very small, very high resolution. Then you need to have a big size. Generally, you have to.
Yeah. Now, so if you begin to reach into that kind of resolution range, typically then you have highly magnified images, and if you have a large complex, then you really have, as you point out, a lot of pixels to deal with. I think in that case, we would have to start looking at segmenting the structure in order to be able to get it into a tractable computational challenge. One thing that has encountered, or that I have thought about, is the fact that considering large viruses, again, if you wanted to determine high resolution structures of those, they carry a lot of redundant information. Typically, they have high symmetry. So there's principally no reason why you should reconstruct the entire redundancy of that virus. You could reconstruct the asymmetric unit, for instance. That would reduce the computational aspect to some extent, but the infrastructure doesn't exist for that at the moment. But certainly reaching towards higher resolutions, especially for big things, would present a memory challenge. In terms of the amount of computations we need to do, I think we could cut sufficient corners to make that a tractable thing. It's mostly about storing that amount of information in an easily accessible format for fast processing. So it's really a data movement and memory management issue rather than a computational one as such. Okay.
I go back to the electron microscopy. So you in your like introduction, you mentioned that different contrast in the image. So what kind of contrast are you using for the single particle analysis?
So single particle analysis always uses phase contrast for its reconstruction procedure. There is a measure of amplitude contrast, but as far as I understand our reconstruction procedures, that's not coherently used in the averaging process at all.
Okay. So with the phase contrast, you have like certain limitations, like you can wake phase object. So you discuss about the phase object. What is the do you apply the phase object no matter how large the particles, independent of the size of the particles you are using? For example, now we're talking about like 100 nanometers or even larger, right?
Yes. Yes, at the moment we are agnostic about what type of sample it is. These approximations are applied without really considering how thick the samples are or whether or not it should work. This is again one of the cases where I've tried to understand exactly why things work better than you would anticipate that they should. Mm-hmm. The phase object approximation that we employ typically breaks down at very thin sample thicknesses, but we tend to use much thicker samples than is compatible with that, but it nonetheless works in our reconstruction protocols.
I guess that is also valid for the multiple scattering.
Yes, the multiple scattering is, yeah, the multiple scattering is different from the amplitude contrast, but I assume that it contributes an incoherent contribution. So it doesn't interact coherently with the unscattered beam, and thus it's not a type of phase contrast that our equations are suited to deal with. And since it doesn't, it contributes incoherently and we don't see it.
Actually, what I saw in my multiple scattering, that actually it contributes coherently.
Does
so
it?
it's yeah. So it's depends. They are both parts.
So it would contribute coherently, but to a different information content, right? So it would superpose with the original reconstruction, but it wouldn't contribute something else. It would be a coherent noise contribution, effectively, right?
No, it will change the contrast of the image.
I see.
But it seems that you don't, you're not worried about it for the single particle reconstruction. And so come back to the phase contrast. Of course, the CTF is a very important parameters. And so I really liked this, what you showed, that pre-particle defocus refinement improved the resolution dramatically, right?
Yes.
And so I want to know, so that how did you said that probably the CTF will be different, depends on where the particles is located in this ice layer. And did you get this number, the defocus values, to see that if it is sensible, like what will be the differences of the defocus and for different particles?
So your question is, how different can the defocus values be for particles within the same ice thickness?
Exactly, within the same picture, yeah.
So typically they can be as thick as the ice sheet that we are using to image. Whether or not that is correctly estimated by this per particle defocus estimation is really sample dependent. So what we are looking for when we're doing this per particle defocus estimation is an agreement between the reconstruction with the applied theoretical CTF and the actual image. So we apply a number of different theoretical CTFs and we look for agreement. Significance in that fit relies on the information manifesting in the CTF, both in the image and the reconstruction being correct. So based on those two things, you would have to assess whether or not you trust that per particle CTF estimation. And typically, you can see that if you tilt the sample and acquire images of conventional test cases, such as apoferritin or beta-gal, then if you do a per particle CTF correction with the nominal resolution you tend to get around 3 angstroms, that produces significant improvements, yes, because you have significance to extend your CTF information and find a better defocus estimate per particle.
So how for the CTF, you always have the tone ring, right? Tone rings, and you have the zero crossovers. How do you take care of the zero crossovers where there is no information?
How do we take care of the zero crossovers in the reconstruction or in the per particle CTF estimation?
In the reconstruction.
So because we utilize many different defocus values, that's not strictly an issue, because we will have zero crossovers that won't sort of contribute any information to the reconstruction. But the probability that those crossovers superpose for all our particle images are fairly unlikely. So typically, we don't get full information from any one particle, but the contribution of all particles together make it so that there's always a number of particles contributing at each spatial frequency.
So you really hope that so this will be focus will be different, so that will be averaged.
Yes, you have to take some care to make sure that you have a defocus range in your data set. But typically, this has never been an issue, especially if you do per particle CTF correction. Then the likelihood that your particles would end up at exactly the same defocus value or have mutually the same zero crossovers is fairly negligible.
Yeah, then I come back to the CTF. So of course, we talked about the resolution. You said probably you won't get a very high resolution data because of the flexibility of the molecules. And what is the impact of CTF on the resolution? Is it any?
So
Like
if
it looks like. Yeah. So sorry, like when you made this pre-particle refinement, you really see the improvement in resolution. But is that the experimentally the CTF will affect the resolution?
So there's a couple of underlying factors that determine what the CTF looks like. Fundamentally, of course, it's an oscillating function, and that
and
oscillation.
there's a damping also, so it's
exactly yeah. No. But principally, the oscillation is very dependent on an exact defocus estimate. So if you get that defocus estimate wrong, as you move to higher resolutions, you will get progressively worse corrections. And to some extent, it will be counterproductive, right? So it's really important that you get an accurate defocus estimate. But as you point out, there's also a dampening function that's dependent on the applied defocus. So typically, you both want an accurate estimate and you want to keep as low a focus, or you want to keep the absolute value of the defocus as low as possible in order to have this dampening and not kill your higher resolution data effectively.
Yeah, we come back. So with all this, I would like to say that actually want to go back to the question we already talked about, like if single particles will replace diffraction or crystallography. Yeah. So if you compare you said no, that so you think the X-ray diffraction still has advantages, but what will be the limitations of the single particles if you really want to go further to like replace electron crystallography? Because so now it certainly there is a very important advantages is that you can really don't need crystals, but in terms of the speed, in terms of the method itself. So can you say something like if we want to further develop, so when the crystallography will be limited, the application of crystallography?
So the first thing I will say is that they are extremely complementary in the sense that as soon as you move towards smaller things that cryo-EM can't handle, biomolecules tend to be easier to crystallize. So they're extremely complementary, and I don't foresee that happening. But in a theoretical case where you would like to, let's say cryo-EM had benefits at smaller molecular sizes that are currently advantageous to use crystallography for. Let's say that cryo-EM would be better to use. What would we need to do to get there? Is that your question?
Yeah.
Yeah. A lot. I don't know that it can reach that far. I don't know if we would have to go to specifically high radiation doses in order to get contrast for such small things, and whether or not they are less prone to radiation damage. That could be one intersection that could make such a thing possible, but that's a fairly straightforward thing that we could do already. In terms of what we would need to do in method development to enable them, I'm not sure. The challenges that are facing small particle cryo-EM at the moment are whether or not you can find significance in your alignment. If you can't, then it's prone to overfitting. If you could reduce that degree of overfitting, you might be able to loosen the regularization and have more significance in fairly difficult to align cases. That could be something you could try. But as I say, I don't foresee this to be a reality, but it's an interesting hypothetical.
Yeah. Finally, so I have a question about like you said in the you have been involved in the developing the Reliant 1 and or 2 and 3. What will be the next version?
what features
Yeah,
are on
exactly. So if you want to further continue.
So I mentioned a few aspects that I think cryo-EM should develop towards in my thesis and in my talk. And I think those should be prioritized, but I don't strictly, I'm not directly involved in the method development of RELION any longer. And the parts that I know are being developed are focused on better interpretations and reconstructions from the same data to basically regularize and filter the reconstruction so that you get better interpretations and better manifestations that are more biological in a sense, to make RELION aware of what a biological sample looks like in order for it to accommodate that type of information. Apart from that, I don't really know what's on the horizon for RELION, but I can very well imagine that we would need to make it faster on more sizable hardware, cutting the corners that I was discussing with regarding orientation and so forth, to make sure that we don't use computational resources in order to process the same tasks that we do currently.
Oh, final question. So about your work, what you are mostly proud of?
I mean, the work that I did together with Dori to make RELION faster has helped so many things, including myself in the other projects that I showed. So it's very easy to be proud of how much we've enabled people to try their own things and discover things that they wouldn't have seen otherwise. I think that's by far the most thing.
Which I agree also. Yeah, congratulations. Yeah. I'm happy. I'm fine.
Actually start my own video. See me and the vast audience behind me. Very good, thank you all. So we move to, do we have any questions from our, let's start first with our physical two-membered audience.
I just want to, may I add one final comment, Pia?
Yes.
You know, a couple of the members of the committee brought up this question about X-ray crystallography versus cryo-EM, and I just want to say that what Björn said is exactly right. I think it's this question of whether cryo-EM replaces crystallography, I think is not really the right question, because they each provide different kinds of information and they each have their strengths and weaknesses. So I think the the answer that he gave, I would just say, is exactly spot on. And one shouldn't view these view one as a replacement for the other, and it's much more relevant to look at these as complementary methods that we will always use, along with lots of other methods as we look at biology. Maybe it's obvious, but I just thought it's worth mentioning.
Thank you. Yeah, good. So, do we have any further comments, questions? Not from you. So, anybody online, please raise your hand or speak out. Let me see if we have any. I don't see anybody. I see a lot of people, but they're not raising their hands. Do you see anyone?
Nope.
Okay. So, last chance. You want to contribute to the discussion?
You can just post it to YouTube.
If not, then I will actually close the open part of this dissertation. Stand up when I do this. We close the open part of this dissertation, and the committee and the opponent, you leave this meeting and you join the other meeting details that I sent you, and we'll meet in 5 minutes. And then we come back here to this room and to this Zoom room when we're done. Okay? Thank you all very much. Let's have a big applause.
Che bellino. Björn! Det kanske... Why?
Yeah. Unable to start video. That's strange.
Ah, it's the host that has stopped it.
Yeah.
Yes, yes, Martha.
Ciao!
Hello.
Du är den bästa.
Björn was muttering comments about some sort of cryo facility at Science Lab, and what he really thought about this. We should mute that.
Men vi kan inte... We cannot switch on our video. They host us this...
You can probably turn on your video.
Yeah,
Jag
but
här med Karin. Vi har mikroskopet.
go into the Zoom room. No, is that not the case?
You're right. Admit all. There we have the thesis committee again. And now we'll just see if Pia comes too.
Yeah.
Ja, på plats, eller hur?
It's a good time for you to mute, Martha. And then, yes.
Okay. So we're back. Let's see. Do we have? I told the committee. The committee was going to come back too because they wanted to hear the announcement and to. And they no, I don't have the link anymore.
You should be able to check on my computer. We have Shadong, Sriram, Otsan.
Yeah, that's great.
Let's see if we have
Oh.
Suparna.
Suparna also. Do we have Suparna?
Yes, we have Suparna there too. Good.
Very good. So then, because normally the head of the committee would do this, but since they are here somewhere,
Yeah.
are they there?
Somewhere in the
Then
middle.
it's, where's Erik? Then it is my great pleasure to tell you that you passed. Yeah. And if I was allowed, I would give you a hug. But it's all also virtual, 2-meter hug. And also congratulations. You did a great job. I think everybody thought so. At least that's what I heard. And well-deserved PhD. Yeah.
Thank you. Thank you.
Woohoo!
Congratulations.
This is weird.
Yes.
Yes. And again, if we have
Björn, congratulations.
this.
Yes.
Let's go.
Björn, great job, Björn. Congratulations, and wish you all the best.
Thank you.
All the best in your career.
Yeah, thank you for a wonderful discussion. It was very, very interesting.
Thank you.
Very broad.
Yes,
Covered everything.
I like that.
Almost.
Yes.
Very good. So
And now's the time to unmute everybody in the Zoom room so everybody can chat.
Yes.
exactly.
There you go. Thank you. And I'm gonna stop the live stream in a
Because-
second too.
I wish we could see Sheldon because she actually had a glass of something. So I can't see her. Come
Let's
on.
see.
Where are you?
You can,
That just seems so nice. You can do gallery view and then everyone can turn on their camera.
Yeah, but then you should do gallery view from your screen.
Oh, I see.
There.
Thanks a bunch, too. There we go.
Very good. So, our enormous audience.
Congratulations.
Oh, you have.
I have. I can take two.
This is real stuff, Erik, or
It is really champagne, so that's the one advantage of only needing to buy sparkling wine for 5 people. I splashed a real champagne for once. Björn,
Yes.
congratulations.
Thank you.
And thanks for a great job. And to both Sriram and the thesis committee, thank you so much for all your help. Cheers.
pleasure, yeah. Cheers. I have my morning coffee, as you can tell.
Yes.
Is it finally morning there now?
Is it? You can tell from the sun is coming up. Yes.
Try to keep my distance. Can we do the proper? We did
Ja,
it.
Hurra! 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 Hurra!
Go on. Thank you so much.
Goal.
You could have been a little bit more hefty.
Well, what can you do?
Yeah, would you take a